Here is a combined summary of the two pages. Inferring causal links or subgraphs corresponding to a specific phenotype or label based solely on measured data is a challenging task. While graph neural network (GNN) explainers have shown potential in subgraph identification, existing methods often offer associative rather than causal insights. To address this issue, the authors propose a novel method of causal link subgraph inference, called CIDER, Counterfactual-Invariant Diffusion-based GNN explainer. CIDER is a model-agnostic and task-agnostic framework for generating causal explanations based on a counterfactual-invariant and diffusion process. It provides not only causal subgraphs due to counterfactual implementation but also reliable causal links due to the diffusion process. Specifically, CIDER is formulated as an inference task that generatively provides two distributions of one causal subgraph and another spurious subgraph. The CIDER framework is further modeled as a diffusion process to enhance reliability, allowing for the explicit quantification of each subgraph's contribution to a phenotype label in a counterfactual manner, representing each subgraph's causal strength. From a causality perspective, CIDER is an interventional causal method, different from traditional association studies or observational causal approaches, and can reduce the effects of unobserved confounders. The authors evaluate CIDER on both synthetic and real-world datasets, demonstrating its superiority over state-of-the-art methods. Traditional GNN explainers provide explanations at a model level, explaining the association between subgraphs and the specific task GNN, but fail to answer the important question: which part of the graph results in such a label? Existing methods, such as 1 and 2, estimate the contribution of each subgraph to the trained GNN's output but may fail to find the causal subgraph. Others, like 3 and 4, aim to provide causal information but ignore the dependence between edges or causal factors, resulting in poor results in low-sparsity situations. In contrast, CIDER addresses these limitations by providing a causal subgraph from data, answering both which part of the graph causally affects a sample's label, and which part of the graph results in the trained GNN's output? CIDER, a novel framework, provides causal explanations from subgraphs to labels or phenotypes by training a graph neural network (GNN) solely on high-dimensional measured data. The framework divides the whole graph into two subgraphs: a causal subgraph that affects the label and a spurious subgraph that does not. CIDER directly predicts the distributions of these subgraphs, enabling the estimation of causal strength, i.e., each subgraph's contribution to the graph's label. The framework models the problem as a variant inference task, predicting the two subgraph distributions using a VGAEBASED framework with two channels. The two distributions are then used to reconstruct the original graph structure, and the contribution of each subgraph to the label is computed by performing sample counterfactual implementation in the spurious subgraph. CIDER adopts a diffusion process to enhance the inference capability, where denoising and distilling the spurious subgraph are performed at each diffusion step. After T steps, each step's causal subgraph distribution and noise distribution are obtained. The noise distribution's significant attribute and counterfactual invariance make CIDER trainable, resulting in a sparser and more reliable causal subgraph distribution. CIDER provides not only a causal subgraph due to counterfactual implementation but also its causal links owing to the diffusion process. Experimental results on both synthetic and real-world datasets, including single-cell RNA-seq datasets, demonstrate CIDER's advantages over state-of-the-art methods. Theoretical results of CIDER ensure interventional causality, inferring the causal subgraph against an output label or phenotype of a sample based only on data. The conceptual illustration is given in Figure 1. A random graph is defined as G equals X, V, E, where each node has a d-dimensional feature vector, and X is a V times D matrix representing the measured data of a sample or graph. The goal is to derive the conditional distribution of the edges, P, E, X, V, from the data. In the context of the synthetic BA2 motif dataset, CIDER demonstrates exceptional performance, achieving nearly 100% accuracy in classifying graphs based on the attached motifs. This remarkable level of precision underscores CIDER's effectiveness and power in addressing complex pattern recognition challenges within graph data. The model's performance on real-world datasets, such as MUTAG and NCI-1, also outperforms all baselines, indicating that CIDER can learn the causality from the subgraph to underlying phenomena and use fewer edges to predict the ground truth. This is achieved through a two-channel VGAE model, which fits P, EC, T-1, EC, T, and simultaneously infers P, S, T, EC, T, allowing for the explanation of both the GNN output and the underlying causal factors. The model's ability to accurately classify graphs and predict ground truth labels in real-world datasets further highlights its capabilities in identifying and explaining causal relationships within complex graph structures. The CIDER method is applied to real-world RNA-seq data analysis, demonstrating its potential and adaptable utility in identifying underlying biological mechanisms. The COVID-19 SCRA-seq dataset, comprising 8,361,000 single cells from 124 individuals, is used to evaluate CIDER's performance. Initially, a graph neural network, GNN, is employed to construct a gene network with 370 nodes and 498 edges, achieving a classification accuracy of 0.85. After applying CIDER, the network is significantly streamlined to 30 edges, maintaining a classification accuracy of 0.82. This result indicates that CIDER preserved approximately 96% of the performance in deducing genogene causal subgraphs while reducing the network's complexity. CIDER is further validated using the COVID-19 RNA-seq dataset from blood, refining the top 1% of highly variable genes, 375 genes, and selecting the top 30 genes with the highest causal association scores for biological analysis. The string network reveals that these 30 genes are partitioned into 3 independent sub-networks, potentially indicating distinct network functions. The isolated COTL1 gene may have a unique impact, with previous studies reporting its role in the immune response and involvement in the superpath's innate immune system. The KEGG pathway enrichment results reveal roots including ribosomes, apoptosis, tuberculosis, and COVID-19, suggesting a direct connection between the key genes and disease mechanisms. The gene ontology and UniProt keywords enrichment results further support the effectiveness of the CIDER method in identifying COVID-19-related genes, emphasizing their role in inflammatory conditions and potential prognostic value for severe cases. The downregulation of the COTL1 protein in the airway mucus of COVID-19 patients is also noted. Overall, CIDER demonstrates its capability in identifying causal genes and cell-type networks, facilitating the identification of underlying biological mechanisms at a molecular level. Here is a combined summary of pages 9 and 10. The authors investigate the application of the CIDER method in identifying genes associated with COVID-19 and acute myeloid leukemia, AML. They highlight the significance of the NOX2 gene, which has been linked to thrombotic complications in COVID-19 patients, and the ribosomal protein family, which has been reported as a potential inhibitor of COVID-19 replication. The S100 calcium-binding protein family is also mentioned, which has been implicated in immune-related keywords and has potential applications in predicting disease severity. The authors then apply the CIDER method to the TCGAL-AML dataset, which includes gene expression profiles from 179 individuals with acute myeloid leukemia. They use the string database and correction networks to construct the original gene network, and then apply the CIDER methodology to identify and refine the top 1.5% of highly variable genes. This approach results in the selection of 51 genes with the highest causal association scores, maintaining an 88.6% accuracy compared to the original gene network. The authors perform further biological analysis on these key genes using tools such as David and Strain. They generate network subgraphs, which illustrate the division of the 51 key genes into 2 distinct clusters, revealing potential unique functions and interactions within the molecular network related to acute myeloid leukemia and ribosome. KEGG pathway enrichment analysis reveals a direct connection between the key genes and leukemia mechanisms, including pathways in cancer and cancer-related signaling pathways. Gene ontology biological process enrichment analysis and gene ontology cellular component enrichment analysis are also performed, revealing enrichment in biological processes such as regulation of apoptotic process, regulation of programmed cell death, and cytoplasmic translation, as well as cellular components such as ribosome, phosphatidylinositol 3 kinase complex, and cytosolic small ribosomal subunit. These findings suggest that the CIDER method effectively filters and identifies key genes with causal associations with acute myeloid leukemia, offering a deeper understanding of the disease's pathophysiology and contributing to the identification of potential targets for therapeutic intervention. The authors conducted enrichment analyses to investigate the connections between key genes identified by CIDER in the TCGAL-AML dataset and related terms. The results showed strong associations with leukemia, particularly acute myeloid leukemia, AML, and the pathway in cancer. Notably, 15 genes were linked to colorectal, gastric, and breast cancers, which are known to increase the risk of developing AML. The string database generated network revealed two significant subnetworks, one related to AML and the other to ribosomes, suggesting a potential relationship between AML and ribosome dysfunction. The authors highlighted the importance of p53 pathway dysfunction in AML and identified RPL26, a ribosome gene, as a critical player in affecting p53 induction. RACK1, another ribosome-associated gene, was found to promote AML cell proliferation and chemotherapy resistance. The ribosomal protein S3, RPS3, gene was also implicated in AML progression, and targeting RPS3 could be a potential therapeutic strategy. The focal adhesion pathway was identified as a significant player in AML progression, and targeting this pathway with specific inhibitors has shown promise in preclinical studies. The authors also discussed the role of KIAA1549L and CPNE8 genes in AML development. KIAA1549L was implicated in the development of acute leukemia through its involvement in fusion genes, while CPNE8 was found to negatively regulate AML cell proliferation. The enrichment and biological analyses of key genes from TCGAL-AML data using CIDER provided significant insights into the molecular underpinnings of AML and related leukemias. The authors introduced CIDER, a new method for causal subgraph inference, which leverages both counterfactual and diffusion implementations to provide generalizability in evaluating a subgraph's causal impact on labels. CIDER can reduce the influence of unobserved confounders, a major advantage of this work. However, there are remaining issues that need further improvement in this framework, including handling unobserved confounders. The paper introduces CIDER, a novel framework that leverages graph neural networks, GNNs, to identify and explain causal subgraphs in complex systems. This model-agnostic and task-agnostic approach is designed to differentiate between causal and spurious relationships within the data, thereby enhancing transparency and trust in AI models. The proposed method utilizes an additive noise model and counterfactual invariants to infer the causal subgraph by training the GNN on measured data. The framework is based on the concept of random graphs, which are used to analyze complex networks in various fields. In the context of machine learning, random graphs can model and analyze large-scale networks such as social networks, biological networks, and the Internet. The paper focuses on identifying the conditional distribution of edges, P, E, X, V, where E and V represent the number of edges and nodes, respectively. To address the problem of causality, the authors formulate the issue as a causal effect problem, with a focus on the causal relationship between the causal subgraph E_C of E and the label Y. The aim is to learn a function f that gives us the causal subgraph E_C. The paper also introduces the concept of spurious subgraph E_S, defined as E_C, and formulates the problem using equations 5 and 6. These equations involve sampling a new spurious subgraph E_S from the marginal distribution P, E_S, E_C, without affecting the GNN's output, thereby making the inference from association to causation. The paper highlights the importance of explainability in fostering transparency and trust in black box AI models and aiding researchers in comprehending underlying phenomena and uncovering intricate network-level patterns. It underscores the significance of understanding the why behind a phenomenon, i.e., its underlying causal subsystem or subgraph, particularly in fields like biology and physics. The proposed CIDER framework offers broad applicability across various domains and has the potential to alleviate unobserved confounder effects, providing a promising avenue for future research. This section of the research paper discusses the use of an additive noise model, ANM, and a structural causal model to understand the relationship between signals and their noisy observations. ANMs are widely used in various machine learning fields, such as image processing and audio processing, where a signal of interest, X, is corrupted by random noise, N, resulting in the observed signal, Y equals alpha X plus N. The presence of noise is essential in ANMs, as it enables the identification of which signal, X or Y, is the cause and which is the effect. To identify the subgraph EC that has the most attribution to a given label yin a graph G, it is necessary to assess the causal effect of EC. However, this requires intervention in S. To tackle this challenge, the authors adopt an ANM to formulate the problem, as depicted by EQs 7 and 8, where E, Y, and Z represent edges, graph labels, and hidden variables, graph representation, that generate E, respectively. Z cans, ZC are sampled from different normal distributions, and FEI's VGAE decoder. Following 66, the task-agnostic phi can be viewed as message passing. The aggregation process essentially acts as a sequence of transformers based on the edges in the feature matrix X. This perspective allows us to form the problem as an out-of-distribution, OOD, detection task. We treat each edge as a transformer applied to X, and aim to identify a set of edges that can be removed without making the resulting representation phi GC, X, deviate from the original distribution. In other words, the phi GC, X, should be a counterfactual invariant representation that only is sensitive to changes in the distribution of edges deemed causal, EC, not those deemed spurious, S. This concept is illustrated in Figure 4a, which depicts our method's directed acyclic graph, DAG. Note that ZC and Zs may exhibit correlations that are not considered here. Figure 4b further demonstrates how our approach integrates with the decision region framework discussed in section 5.1.2. In order to quantify the causal effect or strength of EC, we need to perform an intervention. However, we cannot directly resolve the dependence between subsets of edges. Thus, we cannot use a mask on the original graph G, as conducted in previous works, 1, 67, 72, or calculate the causal effect of each edge separately, 3. Instead, we use the framework of random graphs to solve this problem at the distribution level. Although a graph may not look like other structured data, such as images or lists, it also has distributions. P, E, P, EC, and P, S, represent the distributions of E, EC, causal effect on Y, and S, no effect on Y, EC, respectively. The relationships between them can be formulated by EQ 9. As mentioned in section 5.4, EC is a set of edges, which induces a series of transformations resulting in OOD of phi GC, X, when their distribution changes. Therefore, we can use variational inference models to fit the distributions of S and EC, and obtain the subgraph of E that contributes the most to the label Y. We can use a VGAE-like model to infer the distributions of hinders, even if we do not have access to their actual distributions or samples. Using the counterfactual-invariant representation, we can obtain a partial distribution of S without calculating the maximum likelihood of E-core S. Our goal is to ensure EQ 10 satisfied. By doing so, we can ensure that the set S-hound in Figure 4a has no causal effect on under the condition EC. Although we cannot obtain the total variation distance in EQ 10, we approximately minimize the L1 distance of the due distribution, i.e., the distribution after DA calculus, and origin distribution on R to solve this problem. In other words, our goal is to minimize EQ 11. NC and NIST note the sampled numbers of the causal and spurious edges, respectively. The CIDER framework, a two-channel VGAE, is utilized to simultaneously infer the causal and spurious subgraphs from gene cell expression matrices. It operates on the principle of a diffusion process, where the hidden variable Z is treated as a diffusion process, akin to DALLE2. The model's loss function, LCIDER, consists of three components. L1, which calculates the causal effect of E on Y. LKLD, the KL divergence of ZC and ZS with the normal distribution, and LRE construction, the reconstruction loss of EC union S and E in the form of cross-entropy. Additionally, LTask is used to avoid S always equaling empty set and E always equaling E. The diffusion process in hidden space is characterized by the distributions P, ZCT, ZCT plus 1, and P, ZST, ZST plus 1, which are both normal distributions with parameters micro c theta, ECT, sigma c theta, ECT, micro s theta, ECT, and sigma s theta, ECT. The pseudocode for CIDER, outlined in Algorithm 1, demonstrates the process of sampling ZCT and ZST from their respective distributions, updating ECT1 and ST1 using these samples, and then repeating this process until t equals 0. The analysis process on biological data, as depicted in Figure 5, begins with data cleaning on the original gene cell expression matrix, denoted as Zorigin. Following this, the CIDER model is applied to infer the causal and spurious subgraphs. This process allows for the identification of causal genes and cell types associated with a disease state from scRESeq data. The data cleaning process involves removing cells or genes with zero expression values, resulting in a selected gene cell matrix. This matrix is then used to construct gene and cell type networks, which are essential for CIDER's analysis. CIDER, a tool for causal inference, is employed to generate explanation networks indicating the causal effects of genes and cell types on COVID-19 and influenza. References cited in the paper include studies on graph neural networks, such as GNN-explainer, parameterized explainers, and generative causal explanations. These tools are crucial for interpreting complex models and identifying key factors influencing disease states. The paper also references a blood atlas of COVID-19, which defines hallmarks of disease severity and specificity. This atlas provides valuable insights into the biological processes involved in the disease and aids in understanding the role of different genes and cell types. Other cited works focus on protein-protein association networks and functional enrichment analyses, highlighting the importance of understanding gene interactions and their functional implications. The study also draws on research related to coactosin-like proteins, which have been shown to suppress breast cancer growth and play a role in immune responses. Pathcards, a multi-source consolidation of human biological pathways, is another key reference, providing a comprehensive view of biological processes and pathways relevant to the study. Finally, the paper cites a proteomic profiling study that reveals a distinctive molecular signature for critically ill COVID-19 patients, emphasizing the significance of proteomics in understanding disease progression. In summary, the paper leverages various tools and techniques, including CIDER, graph neural networks, and biological pathways, to analyze scRESeq data and identify the causal genes and cell types associated with COVID-19 and influenza. The findings contribute to our understanding of the molecular mechanisms underlying these diseases and could potentially lead to new therapeutic strategies. The research paper explores the potential role of S100 proteins, specifically S100A8 and S100A9, in the regulation of NADPH oxidase 2, NOX2, activity during COVID-19 infection. These proteins are known for their involvement in various physiological processes and diseases, including inflammation and oxidative stress. The study suggests that S100A8 and S100A9 may play a crucial role in the hyperinflammation observed in severe COVID-19 cases, possibly through the activation of NOX2. The paper also delves into the relationship between calcium levels and COVID-19 severity. It cites studies showing that low serum calcium levels are associated with increased mortality and severity in COVID-19 patients. This could be attributed to the importance of calcium in various cellular functions, including immune response and oxidative stress management. Furthermore, the authors discuss the potential therapeutic applications of plant lectins, which have been found to exhibit antiviral properties against enveloped viruses. These biomolecules may serve as a new strategy for combating COVID-19, leveraging their ability to interact with glycans on the surface of viruses and prevent infection. Additionally, the paper touches upon the status of the complement system and mannose-binding lectin, MBL, in COVID-19 patients. MBL is a key component of the innate immune system and has been implicated in the regulation of oxidative stress and inflammation. The study highlights the potential therapeutic applications of antiviral plant MBLs in COVID-19 treatment. Lastly, the paper mentions the significance of RNA splicing alterations in acute myeloid leukemia and their association with poor prognosis. This section emphasizes the importance of understanding the molecular mechanisms underlying cellular stress responses in different diseases, including cancer and infectious diseases like COVID-19. The research paper discusses the relationship between breast cancer and acute myeloid leukemia, focusing on the role of p53 and ribosomal proteins in the development of these diseases. It begins by citing several studies that have identified a high prevalence of p53 pathway dysfunction in acute myeloid leukemia, which is associated with poor prognosis. This is followed by an exploration of the role of ribosomal proteins, specifically nucleolin and ribosomal protein L26, RPL26, in regulating p53 translation and induction after DNA damage. The paper then delves into the relationship between RAC1 and the proliferation of THP1 acute myeloid leukemia cells, as well as its involvement in chemotherapy resistance in T-cell acute lymphoblastic leukemia-derived cell lines. Additionally, it touches upon the overexpression of RPS3, which promotes acute lymphoblastic leukemia growth and progress by down-regulating COX2 through the NF-kappa B pathway. Furthermore, the paper highlights the potential of focal adhesion kinase as a target in AML and MDS, given its association with enhanced blast migration, increased cellularity, and poor prognosis. The study also mentions the presence of novel fusion genes in pediatric B-cell precursor acute lymphoblastic leukemia and acute myeloid leukemia, such as PAX5KIAA1549L and RUNX1C110RF41. Overall, this section of the research paper presents a comprehensive overview of the complex interplay between various genetic factors and their implications for the development and progression of acute myeloid leukemia and breast cancer. It underscores the importance of understanding the molecular mechanisms underlying these diseases to identify potential therapeutic targets and improve patient outcomes. The references cited on pages 25 and 26 of the research paper span a wide range of topics in machine learning, graph theory, and network analysis. The citations include works on graph-based methods for analyzing networks in cell biology, 60, complex networks from graph theory to biology, 61, and geometric inhomogeneous random graphs, 62. Additionally, the references cover semi-supervised classification with graph convolutional networks, 63, graph attention networks, 64, and the power of graph neural networks, 65. The citations also touch on asymmetry learning for counterfactual-invariant classification, 66, robust counterfactual explanations on graph neural networks, 67, and systematic evaluation of explainability methods for graph neural networks, 68. Furthermore, the references include works on robust learning with the Hilbert-Schmidt independence criterion, 69, certified adversarial robustness with additive noise, 70, and regression by dependence minimization and its application to causal inference in additive noise models, 71. Other topics covered in the citations include interpretable and generalizable graph learning via stochastic attention mechanism, 72, empirical model approach for evaluating pH and conductivity on pollutant diffusion in soil environment, 73, and the potential role of particulate matter in the spreading of COVID-19 in northern Italy, 74. The references also include works on the role of social networks in information diffusion, 75, denoising diffusion probabilistic models, 76, and deep unsupervised learning using non-equilibrium thermodynamics, 77. The citations also cover generative modeling by estimating gradients of the data distribution, 78, and hierarchical text conditional image generation with clip latents, 79. Overall, the references cited on pages 25 and 26 demonstrate a broad range of research interests and topics, from graph theory and network analysis to machine learning and deep learning. Here is a combined summary of pages 27 and 28. The performance of CIDER is evaluated on two benchmark datasets, MUTOG and NCI-1, whose statistics are presented in Table 2. These datasets consist of 4,337 and 4,110 graphs, respectively, with an average of 30.32 and 29.87 nodes per graph, and 30.77 and 32.30 edges per graph, respectively. All experiments were conducted on a server with a single NVIDIA RTX 3080 GPU, 2.2 GHz Intel Xeon Gold 6148 CPU, and 256 GB RAM. The training of CIDER utilizes the Atom Optimizer with a learning rate of 0.001 and weight decay of 0.0005. The batch size is set to 128, and the model is trained for 500 epochs. The number of diffusion steps is set to 10, with a single causal sample and 4 spurious samples. For comparison, baseline models Gene Explainer and PG Explainer are implemented using the PYG version, while GEM and Orphic X employ the official versions provided by the authors. The CIDER framework is designed to address the limitations of existing graph explainability methods, which often rely on heuristic-based or proxy-based approaches. In contrast, CIDER leverages a causal inference framework to identify the causal relationships between graph nodes and the target outcome. The method involves estimating the causal effect of each node on the target outcome using a diffusion-based approach, which enables the identification of causal and spurious relationships. The experimental results demonstrate the effectiveness of CIDER in identifying causal relationships in graph-structured data. The results are presented in Figure 1, which shows the superior performance of CIDER compared to baseline models in terms of precision, recall, and F1 score. The ablation study in Table 3 further highlights the significance of the diffusion-based approach and the importance of considering spurious relationships in graph explainability.